The broadcast is now starting. All attendees are in listen-only mode.
Everybody, thank you for listening in today, Wednesday, April 25th, to our Webinar Wednesday Lifelong Learning Faculty Series. Welcome to today's topic, Lessons in Progress Treating Cystic Fibrosis with Application to Other Childhood Diseases, with Emory's own Dr. Eric Sorscher, pediatric researcher. My name is Mara Felver-Lore, and I'm the Director of Global Alumni Relations at Emory Alumni Association. I hope you'll visit our website and learn about all of the programs we host internationally. These are posted at www.alumni.emory.edu/calendar. You can also find podcasts of all of our faculty lectures on iTunes U at alumni.emory.edu/faculty on iTunes. Now it is my pleasure to introduce our featured speaker, Dr. Eric Sorscher, MD. Dr. Sorscher is currently a professor in the Department of Pediatrics. He has been named Hertz Professor in Cystic Fibrosis Research, along with being an eminent scholar of the Georgia Research Alliance. Dr. Sorscher is originally from Flint, Michigan. He graduated summa cum laude with honors in biology from Yale University in 1979. In 1984, he completed his medical degree from Harvard University. His internship, residency, and fellowship training were at Massachusetts General Hospital. During this presentation, Dr. Sorscher will describe his research and the ways in which a common and lethal genetic disease is being effectively addressed by scientific groups, the pharmaceutical industry, and regulatory agencies in a manner emblematic of drug discovery for diverse human disease states. Along with the enormous progress that has been made over the past few years regarding development of new and transformative therapies for cystic fibrosis, emerging technologies being applied in this area have relevance to many other illnesses. If you have any questions throughout the presentation, please feel free to chime in. We want this session to be an open dialogue between all attendees. We appreciate your understanding if we can get to all of them. At the end of the program, I will share Dr. Sorscher's email address with you. He loves to hear from alumni, parents, and friends of Emory. Dr. Sorscher, thank you for joining our webinar today.
Thank you very much, Mirfel. It's a pleasure to be here, and I appreciate the opportunity to come and speak regarding cystic fibrosis and some of the lessons that have been learned in that disease and the relevance to other childhood diseases as well. Should we advance the slides? Here we are. Thanks. So my interest in cystic fibrosis began when I was a trainee in medicine in Boston in medical school. And I was working in my room, and I decided one night to take my guitar over to the Children's Hospital, to a ward on the Children's Hospital that took care of children and young adults, mostly an adolescent ward called Division 37. And that ward, you really met three kinds of people: people who had inflammatory bowel disease, people with sickle cell anemia, and patients who had this disease, cystic fibrosis, that I had never heard of before. And that experience of going to that hospital night after night, through that experience, I met some of the most creative and brilliant and passionate people that I had ever met. And I decided that this was going to be my passion too. And left medical school for a year to work on CF, and have been working on it ever since medical school and really throughout my professional career. When I was a medical student, people with cystic fibrosis often died in their early 20s. So I met many people during that time who were very ill. And when I was a boy, people with CF typically lived only into the first few years of life, maybe age 4 or 5. Now it's very common for people with CF to live much longer, into their mid-40s, for example. But much more work has to be done, and some very exciting things are happening in CF-related science and research, and that's what I wanted to share and convey with today's audience. And thank you again for calling in and your interest. So there are three topics I want to cover in today's presentation for the alumni. First, I want to talk about clinical features of cystic fibrosis and underlying disease mechanism. And then something that's been called precision or personalized therapy for the disease, approaches to challenges. This is an initiative that really covers all of medical research. For most diseases, there's a desire to become more precise or more personalized so that the therapy that's given to a person will really be tailored to the form of a disease that person has. This was introduced by President Obama at a State of the Union address a couple of years ago when a patient with cystic fibrosis was actually asked to stand. And this new initiative at the National Institutes of Health was undertaken. So what we learn in cystic fibrosis is emblematic of many other diseases, and the concept of precision therapeutics is going to become a common part of our language for medicine. So I thought it would be worth introducing that notion here. And then finally, some future directions regarding CF and other orphan diseases as well. And by orphan diseases, I mean rare diseases. Often they're diseases of children, but it means a disease that there are a relatively small number of people that suffer from the disease or have the disease, and that's the designation that's often given, which is an orphan disease. So I want to start with some clinical features of CF, just to tell you that classically, when someone with cystic fibrosis presented or showed up at the doctor's office, the way they would often present would go something like this. A mother and father would have a child, and the child just wasn't growing very well. In terms of both weight and height, the child wasn't the same as other kids in the family or maybe other kids at school. There was something slow about the rate at which the child was growing. The medical term for that is sometimes called failure to thrive, not a particularly pretty term, but the pediatrician would see the child and say, boy, there's a long list, a long differential diagnosis for things that can cause a child not to grow well, and they include everything from psychological to nutritional to chronic illness and to many, many other things as well that can cause a problem with growth. And one of the things the pediatrician might consider is the diagnosis of cystic fibrosis or CF if a child isn't growing properly. And the test that would be sent, at least classically, was called a sweat test. Cystic fibrosis is the disease where public awareness used to be raised by saying, kiss your baby. And if the baby tastes salty, think about the possibility that the baby might have CF. There's nothing else, with very rare exceptions, almost nothing else known in health that causes the sweat to be so salty as it is in CF. And there were times during heat waves in New York City when the disease was first described when kids would show up at the hospital just flaking the salt off their skin. And if a sweat collection is done on a child who's not growing well and it comes back very salty, then the pediatrician will know with a high degree of certainty that the reason for the failing to thrive is because the child has CF. And the pediatrician will know that in the pancreas, the organ that makes the digestive enzymes that allows proper nutritional absorption and proper growth, the pancreas has been damaged, and that the ducts that carry the digestive enzymes into the intestine where digestion happens, those ducts have been blocked by secretions. Secretions that are so thick that they're sometimes called concretions because to a pathologist, they can look as thick as concrete. And they block these pipes or ducts that carry the digestive enzymes to the intestine, and big fluid-filled cavities fill up proximal or in front of the blockage. And those cavities are called cysts, and the cavities are loaded with digestive enzymes that cause self-digestion of the pancreas or autolysis. So the scarring that results, the fibrosis that results, is where the disease gets its name. The full name of the disease is cystic fibrosis of the pancreas. So fluid-filled cavities of digestive enzymes and scarring, and that's the reason the disease is called cystic fibrosis of the pancreas. Those problems with pancreas secretions and with digestion, those can usually be managed or helped by giving oral pancreatic enzymes, and people can get back on the growth curves and do quite well. There are no cognitive disabilities and no musculoskeletal problems per se in CF. It's really a disease of thick secretions of most of the exocrine glands. And once the problem in the pancreas and the liver have been better managed, because the liver can also have thick secretions, the same kinds of thick secretions also happen in other glands as well. So the major problem in the disease, the major morbidity and mortality, and the major thing you'll be familiar with if you've known anyone with CF or you have someone with CF in your family, will be the thick secretions that occur in the lungs. Those secretions build up, and with time they block the airways, they become infected with unusual organisms, and they cause a lot of inflammation in the lung. And the consequence of these changes is that people with CF have a more and more difficult time breathing until, as one patient described for me, it's like trying to breathe with someone's hands always over your face. And people come into the hospital more and more frequently for antibiotics to clear the infections and for physical therapy, where a parent or a physical therapist will pound on the back of someone with CF to try to help them mobilize those secretions to bring them up so they can be coughed up or swallowed. And that's the life of someone with CF is medicines to treat lung and other exocrine gland health problems. And that's the way we often think of cystic fibrosis, as a disease of the exocrine secretions. This slide, taken from Damjanov's paper in 2000, describes some of the findings in CF. And you can see in this slide the abnormalities of the pancreas, as well as the liver, not shown here, and the lung, and the abnormal sweat, which is very high in, in sodium chloride and salt. Other abnormalities as well. The disease is very common. About 1 in 30 people is a silent carrier for this disease, and if 2 carriers have children, something like 1 in 4 of the kids will have cystic fibrosis. The reason it's so common is interesting. There are other diseases, for example, a disease, sickle cell anemia. It turns out if you're a silent carrier for sickle cell anemia, you will not die if there's an outbreak of malaria, a very common lethal disease in Africa. So in Africa, historically, when waves of malaria would go across the subcontinent, there might be a few people who were carriers of sickle cell disease, and those people wouldn't die of the malaria. Those people would be protected, and those people would live, and they would be enriched and maybe move to another village. And then a few years later, many years later, if another wave of malaria came through, again, everyone would die except the carriers for sickle cell. And as this went on over many generations, the frequency of carriers for sickle cell became much higher. Carriers are more or less asymptomatic. And that's why among people of African ethnicity and descent, now we see a very high carrier frequency for sickle cell in those people. And if 2 carriers for sickle cell have a child, this is another orphan disease, of course, the child will have sickle cell disease. Carriers are relatively asymptomatic. Almost certainly something in European history led to the very high carrier frequency of cystic fibrosis. If you're at the movie theater, it's likely there are a few silent carriers in the theater with you. If you go to a football game, there are many, many carriers sitting around you in the stadium that don't know they're carriers unless they've been specifically tested. And the reason for the high carrier state among people of European ethnicity and descent is because of some infectious disease that must have ravaged Europe over the last several thousand years and led to this enrichment of people who were carriers. People who were carriers did well and survived, and they had an advantage, in fact. But if 2 carriers have a child, that child will have CF. And this is information that's of interest often to families where CF is in the family, because it's an explanation, a medical explanation for the high carrier frequency of the disease. I want to say a few words about what really goes wrong in cystic fibrosis and our best understanding of the mechanism that underlies CF. And in order to do this, I want to show a video that is a microscopic view of what it looks like on the surface of the major windpipe. If you could miniaturize yourself and walk along the surface of the windpipe or trachea and go into the divisions of that windpipe called bronchi, you would see something like this. You would see cobblestones, which are the tops of the cells. And here we're standing. If we were standing on these cobblestones, our feet would be planted on the cobblestones, and we would be standing in the airspace. So air would be flowing this way. And you can't see it now, but when I start the video, you'll appreciate that every one of these cells are small projections. They're like hairs, and they beat very frequently and directionally. And it's the beating of these structures called cilia. This is always going on in all of us, that allows mucus that's in the airways to be escalated to the back of the throat, or it can be swallowed or it can be coughed up. This is called the mucociliary escalator. And you can appreciate here how the cilia are beating. And if I take you a little further into the video, you can see here, these are the cilia. These would be the cells. This would be where the air is passing, and this would be the tissue of the lung. And you can see how the cilia beat. Now, on top of these cilia is always a layer of mucus, and I'll say more about that in a second. And it's necessary for these cilia beating, to properly clear that mucus and to mobilize it. And in order for the cilia to do that, water and salts, electrolytes, have to pass from the inside of the cells of the lung out into the airway where the air is so that these cilia can be properly extended. If fluid and electrolytes don't move, then the cilia can't be properly extended. They're not bathed in water, in fact. And as a result, they bend over, they're no longer extended, they become plastered to the surface of the cells that line the airway, and the mucus no longer moves and it builds up. And this is the reason for the accumulation of mucus in the lungs of someone with CF, the reason that after a time it's like breathing with someone's hands constantly over your face. I want to show that in a little more detail in a video that I had made a few years ago with a professional scientific artistry group that had actually won several Oscar awards. It was an interesting experience because when I was taken to the offices in Bethesda to have the video made by their medical artists, scientific artists, it was daunting. They had their Oscars out in the waiting room, and then when they took me back, it was people in sandals and shorts who just loved their work, and they helped me put together this video that describes what goes wrong in cystic fibrosis. It's something we've worked a lot on in our laboratory, and so I wanted to share it with you. What we're going to do is we're going to take a journey in the artist's conception as if we were doing a bronchoscopy at first. We're going to go back past the vocal cords in the back of the throat and then into the windpipe, into the trachea, and then into one of the big bronchi, another one of the big airways. And then we're going to magnify, and we're going to see what the cells look like the cells I showed you a second ago in that microscopy, but we're going to see what the artist's rendition of these cells look like. And we're going to see the cells with their cilia beating. Over the cilia, we're going to see the mucus being moved. And then we're going to magnify again, and we're going to see proteins that line these that line the airways. And it's these proteins, the proteins called the CFTR, the cystic fibrosis transmembrane conductance regulator. It's those proteins that allow the passage of chloride and bicarbonate and water following passively that allows the cilia to extend normally and to beat normally and to move mucus. And it's those proteins that are missing or are not working properly or absent in cystic fibrosis. I'll show you the video in a second, but it's as if lining the airways, all along the surface of the airways, there are these little portals or doorways or gateways, and they open and close. And when they open and close, when they open, they allow the passage of chloride and bicarbonate and water, which allows the normal extension of those cilia, and then allows mucus to move. So let's then we'll go down into the bronchoscopy now, and you can see we're going to the back of the throat and through the vocal cords, and now we're in the trachea. And the trachea bifurcates, it breaks into two parts, and now we're in one of the main stem bronchi, and we're going to magnify. And this is the picture the artist drew. Just to stop there for a second. The air would be here. These are the cells that line the airway, and these are the cilia. Those extended structures that are always beating, like hairs in our airways, they're always beating, and they beat in a rhythmical and directional way. And this would be a small layer of mucus in someone that doesn't have CF, overlying those cilia that's being moved or mobilized so that it can be cleared. And you can see the cilia beating rhythmically. And now we're going to take another magnification step, and we're going to go between two cilia. And we're going to look here, and we're now between two of the cilia. And this would be the layer, the membrane across the cells. This would be the air, and this is the inside of the cell. And these balls represent ions, electrolytes like chloride, bicarbonate, that are moved through these proteins. These are the CFTR proteins. These are the proteins that are not working properly in cystic fibrosis. The balls would move through these portals or gateways or doorways from the inside of the cell out. And when they move from the inside of the cell out, water follows passively, and that allows the cilia, which are much too large to be seen here, to be extended properly. There are many things that can go wrong with these proteins in cystic fibrosis. We'll say more about that in just a second. But if we think about now the chloride and bicarbonate moving through the CFTR proteins and the water following passively, what that allows is it allows the cilia to be normally extended and normally hydrated, and they're able to beat properly. This is happening in all of us all the time, to move mucus. Now, what would happen if that mechanism were broken? What would happen if, for example, these doorways or gateways or portals that allow the normal movement of ions, of chloride and bicarbonate, what if they weren't working so that nothing could get through? You would see that as a result, there could be no movement of water, and as a result, the cilia wouldn't be able to extend properly. And there are many things that can go wrong with these proteins in cystic fibrosis. These are the proteins that are encoded for by a gene that's responsible for CF. Sometimes the doorway is just simply locked shut, as you see here, so that nothing can pass through. Sometimes the doorway is made so that this portal is made so that it's got a small synthetic defect. And as a result, the cell inside of the cell, it degrades it, it destroys it before it can ever get to the place where it needs to be in order to work. Other times it's made with larger abnormalities, whole parts of the protein might be missing. But if this protein isn't here, the important thing to say is if the protein's not here, no chloride and bicarbonate can move, no water follows passively, and the cilia will bend over, become plastered to the surface of the cells, and no longer be able to move mucus. And that's what we're showing now as we scan back out again. Nothing is moving now because of the defects in the CF protein. The cilia can't be extended normally, no water, no hydration, and they don't beat. The mucus then starts to accumulate, it becomes thicker, and it becomes infected. And that's our best understanding of mechanism of the reason that people with CF get sick. And so you're now at the very leading edge of our understanding of why people with CF have thick mucus in an organ like the lung, why it becomes infected, and what the problem is, the fundamental problem in the disease. Now, there are lots of things, as I say, that can go wrong with that protein, that doorway that allows the chloride and bicarbonate to move and allows the cilia to be extended properly. There are many things that can go wrong. In fact, more than 1,600 have been described so far in different people all around the world. It's a very common disease, as I said. Every 35 or so, 30 or 35 people is a carrier. And because it's common, it's most common serious single gene defect among people of European extraction. Because of that, you can imagine that the approach to therapy can be complex. If there are 1,600 different things that can go wrong, there might be many, many different ways, many different problems that can occur, and many different therapies that can happen. So that if the protein is made and it's locked shut, that might require one kind of drug. And if the protein is made but it has a small defect that causes it to be degraded by the cell, that might cause another kind of therapy. And this is the idea that I wanted to introduce in the next part of the talk, which is the idea of precise therapy or personalized therapy. If you know the person's problem, the problem with their CFTR, the problem with their gateway or portal or doorway, if you know what it is, you can devise therapies that will be specific to that particular problem. And in CF, that's taken on an enormous dimension where the idea of tailoring the therapies to the form of the disease that people have, or precise or personalized medicine, that's the concept that the field has worked on now for many years. And the reason it's important is because, as I say, it's emblematic of every other disease, whether you're talking about cancer and the cause of a person's cancer, or heart disease, or you're talking about diabetes, or you're talking about an inflammatory condition like rheumatoid arthritis or lupus. Whatever the disease is, there's interest in understanding the molecular mechanism of the disease to a point that you can tailor the therapy to that particular problem. And cystic fibrosis is doing that successfully now. And so it's representative of many other diseases. So let me turn now for a moment and talk about precision or personalized therapy for CF and what's been done. This is a pretty technical slide, and I just show it to reiterate a couple points I've already made. One is that if there are 1,600 different things that can go wrong with this is the portal or doorway or gateway that I've described that allows chloride and bicarbonate to pass through, again shown by these balls, and then water follows and allows the normal extension of the cilia. If there are 1,600 things that can go wrong, it's good to group them if you can. And there are 6 classes that have been established for the kinds of things that can go wrong. Different classes, like a channel that can never open, would be handled by a different therapy than a channel that's quickly degraded, or a channel that never is really made very well in the first place. And so the therapies are tailored. And this is the idea of personalized therapies. We have therapies called potentiators, which fix a broken CFTR that's locked shut, and therapies called correctors, which fix a CFTR that's made improperly and is degraded before it can ever get to the surface. These are all called modulators as a group. And the names of the specific things that go wrong with CFTR are of great interest, of course, to patients and families, and I'd be glad to answer questions about them at the end of the talk. But for the purposes of this presentation, as a general introduction to cystic fibrosis and what we know about it, there are many things that can go wrong, and the therapies are being tailored to the things that are the problem. Now, how do you develop a new therapy for a disease like cystic fibrosis? We have a good understanding of what goes wrong, and we've reviewed that, and everyone now is sort of, I think, up to speed in terms of our best understanding of what goes wrong. But what's been done to develop these new therapies for CF is really remarkable. And the reason it's remarkable is because some people are calling these new treatments a cure for a disease that when I was a boy, people would die in early childhood, and when I was a medical student in their young 20s, and now in their 40s, more needs to be done. And the approach that's been taken in order to develop these therapies is really transformative. And again, it has implications not just for CF, but as I'll say in the end, for any disease you might be interested in. If you know that you have a target, like a CFTR gateway or portal that's locked shut, and you know that you can study that in cells, you can develop therapies by what's called high throughput drug discovery. And this is the way this approach works. You take a dish with 380 wells or more than 1,000 wells, and into each well of the dish, you put cells that have a defect that you're interested in. In this case, it would be a problem with that gateway, that doorway, that CFTR portal that I've described, that protein that allows balls of chloride and bicarbonate to move through. And every well has cells in it, and every cell has a gateway, a doorway, a CFTR that has a problem. Say it's a doorway that's locked shut. And then you engineer the cell so that if that doorway ever opens, the cells will become incredibly bright. And if a robot then looks at them, it'll be able to find wells where the cells have become incredibly bright. Again, the reason this is important is because it's not really at all CFTR specific. It can be applied to infectious diseases, inherited diseases, cancers, and other conditions as well. You give this dish, which has 1,000 wells or 400 wells, say, to a robot. And the robot looks at every well and says, none of those wells are bright in CF because every portal, every doorway is locked shut. But the robot has a library, an unusual use of the word library perhaps, but a collection of 1 million random drugs. And it takes drug number 1 and it puts it in well number 1, and drug number 2 and puts it in well number 2, and drug number 3 and puts it in well number 3. And it gets up to, say, 384 wells in this plate, and then it loads it up on this apparatus, and then it takes another drug. And then it loads another 300 or 1,000 drugs in the next dish, and another 300 or 1,000 drugs in the following dish, until it screens over the course of several weeks 1 million drugs. And it comes back after a couple days and it says, are any of these wells now very bright? Have they lit up? Because if they light up, we don't know why, we don't know how it worked, but we know that that drug was able to open a doorway that was locked shut. And maybe in 1 million drugs, you'll find 8 or 9 or 10 that really open the CFTR ion channel that's locked shut very effectively. And those drugs then become candidates for therapy. They move forward through the usual routines. They go to animals to make sure they're safe. They go to humans to make sure they're safe. They go to humans who are sick, and phase 3 trials, and ultimately they go to patients. And we now have drugs that work for many patients with CF, as I'll say in a second. And some people are calling these potentially cures for the disease. They haven't been around very long, so they have to be around for a much longer time. And until we know more about them, we really can't say that about them with certainty, but they clearly make a big difference in the health and well-being of patients. When you give a patient, for example, who's got a channel locked shut, one of the drugs, within a couple weeks, their sweat chloride is normalizing or normal. And there's been nothing in 50 years that's been able to correct that problem in people with CF. Lung function also improves very rapidly. So within a couple of weeks, there might be a significant improvement in how well they can breathe. And so this kind of approach, high throughput robotic drug discovery, and this is an example of a robot here, is emblematic of what can be done in many diseases. And it's being done to great effect in cystic fibrosis. It's been in the news a lot, both in the lay literature and also in some very good scientific journals. The Cystic Fibrosis Foundation, which funds a lot of this research, has been really visionary in thinking about what needs to be done to help people with the disease, and it's been exciting to see the progress they've made. A drug was just approved, a new drug was just approved a few weeks ago for a large number of patients with CF, and if you look across the board, and again, the forms of CF are less important here maybe than the percentages. There's now a drug that will reach 46% of people and another drug for 12%, and it's been projected that another 36% will be reached within the next few years. And at that point, we'll have an effective modulator type treatment tailored to some extent to the kind of disease that people have, developed through this technology of high throughput robotic screening that will benefit many people with the disease. And I've worked on the disease for many years. There's never been a more exciting or an optimistic time in the history of this disease for people that have cystic fibrosis, because more and more people will have these drugs available to them. It's been very successful. It's important to say also, though, that there still will be something like 5% of people that won't have a drug because there are so many different forms of cystic fibrosis, so many different mutations that can happen in the gene. And that has become a major priority in our own laboratory and in laboratories of people around the world, and of course for the funding agencies like the CF Foundation and the National Institutes of Health to make all of this research possible, as well as for the pharmaceutical industry that's developing the drugs. Everyone is strongly focused on coming up with better ways to help those people that don't yet have a drug when these new drugs become available. Again, it's a very exciting and promising time just because of the success. And I want to emphasize in this slide from the CF Foundation that the pipeline is enormous. These are the approved drugs, and these are the drugs that are in testing or entering testing soon. And this is part of the reason for the optimism. There are many options. And so for people that have a rare form that do not yet have a drug, those will be evaluated by these new compounds and then new projects starting all the time to try to use this technology to help people with CF and ultimately with other orphan diseases of children and adults and more common diseases as well. There are some important challenges to what's been done so far. As I mentioned, the first of these drugs was just approved in 2012, and people will need to be on these modulators in concept at least for many years, for the rest of their lives perhaps. And so we need more experience with the compounds to really make sure that they don't have any sequelae, that they really are safe. And everything looks positive and promising now, so it is an exciting time. Another thing to say is that sometimes a mutation in the CFTR protein in that gateway or portal that I've shown you, sometimes that mutation can be very complicated. We think of tailoring the therapies to a specific problem, like that channel I showed you in the video that was locked shut, so no ball can get through. And we want a drug that'll open it up. But for many mutations, among the 1,600 that can happen, sometimes there's more than one problem. And so the idea of really tailoring things carefully isn't really as important as just finding the ones that will work. And so that's one of the challenges, is just the complexity of some of the problems we see, and the way we're overcoming that, the way we're addressing that is being a little less precise. Just simply asking which of the drugs that we have will work for you, not so much what's your mutation and what's the precise mechanism. There are very large numbers of mutations. Some of them are very rare. And this is an important thing to point out. It's been estimated there may be 1,000 different forms of the disease for which there are 5 people or less each. And you know, to get a drug approved for a form of a disease, classically it requires a phase 3 double-blind placebo-controlled trial at the FDA. But if there are only a couple people who have it, that's not possible. So I'll say in a minute that there are creative ways that this is being addressed and that we can get around this problem. And then finally, the modulators that we have so far are expensive. And this has caused a problem with access in some settings, and that has to be addressed as well. This is just to say that one way that we're addressing the issue of a lot of different kinds of mutations and which drug will work for whom is through this idea of what's been called theratyping. There's a major initiative being undertaken at some drug companies and also at the Cystic Fibrosis Foundation, where hundreds and hundreds of mutations, including very rare ones like this one, where this particular mutation I think is present in 5 or less people in the world, where we're looking at the drugs that are approved and we're asking, do these drugs work? And we're finding that they do. And so those patients may become candidates. And these are the 2 of the approved drugs. And you can see that there are many mutations that have been already approved for these, and across many different kinds of classes of problem, but more needs to be done so that drugs will be available to everyone, so that no patient with CF will be left behind and not have a drug. And again, that's an emphasis in our own lab. One can argue that, like more common CFTR defects, the really, really rare ones, they often don't have just a single problem. And that assertion has implications with regard to the precision of the medicine we can give. As I've said, we still need to know what the problem is, and we still need to understand the mechanism. But what's most important is we have drugs that are active, and we need to screen those to see who they will help. And the word that's sometimes applied to that is stereotyping. Which of the types of CF do the therapies work for? So now I want to conclude just by saying a few words about some future directions for CF and other orphan diseases. One thing that's going to become more and more important is whether, if we have a patient in a remote area and they're the only patient in the world who has this, say, a particular form of CF, can we use something in a test tube to predict whether or not they'll respond so that we can get them the drug and they can use it? And there are a number of different systems. Cell models, something called organoids, biopsies of the intestine, cells that are taken at lung transplant, progenitor cells that are differentiated so they look and act like lung cells. Many of these we're working on in labs all over the world. People are using these systems so they can take a person with a really, really rare form of the disease and predict whether or not they'll respond. These are just some of the models that we've established for this purpose. And the FDA, and what really was a transformation, has announced recently that this kind of data, in vitro assay data, can be useful in their decision-making process about which drugs will and will not be approved for a certain form of the disease. Another point I want to make is that even when you know the form of cystic fibrosis someone has because you've looked at their DNA and you can understand it, and of course now most patients are diagnosed at the time of birth so that a healthy baby will be born. It looks like a healthy baby, and then a genetic test will be done and you will learn that the baby has CF. It can often be even before they're sick. But when you look at the kind of CF they have, a common kind or less common kind, even people who have the same mutations in their portal or gateway in their CFTR protein, they can be very different in the way their disease manifests, how they do, and in how they respond to these drugs. And so there's a great deal of interest in being able to really be precise. And one of the things we have to understand, these are pictures of it's just a representation of the length of that protein in its genetic form, in the DNA form. And this is the information for making the protein. And each of these is a different person. And each of these different people, none of them have CF, but each of them you can see every time there's an upward tick, this is a different small change, a polymorphism it's called, or a SNP, somewhere in their CFTR. So the NIH has run a program called the 1,000 Genomes Project, and if you look at these just across the part that represents the information for making the CF protein, you can see there's an awful lot of differences among different people. We have to understand that better. And we have a faculty member at Emory who's devising ways to get at that information so we can really be precise in our laboratory and others about who will and won't respond to drugs. Another very important technology for the future is to say something like this. Look, we know the problem. The problem is with that portal, that gateway, that CFTR protein that I've been showing you, and we just need to fix it in the lungs. So let's find a way to take the gene, put it into a virus, or in this case into a nanoparticle, which is just a soap bubble that has some protein in it that can be used to deliver to those cells where the gateway is not working properly. Let's deliver it. And this has been called gene therapy. And in the next several years, we will see more and more examples of orphan diseases and other diseases where gene therapy has been successful. Recently, for example, a group in Pennsylvania has devised a way that people that have a certain rare form of blindness can be treated with a gene therapy, and that blindness can resolve. It was an interesting story. They did this first in dogs, and they showed videos of the dogs bumping into things and crying, and it was sad. And then the dogs, after the therapy, being able to see, and they took the dogs to Capitol Hill and marched them around Capitol Hill, and now they have an approved therapy for a very rare form of blindness. So what can really be done is remarkable. In many of the common inherited diseases of liver is a relatively straightforward place to put a gene if it's defective. The surface of the lung is harder, but we will see more and more of this in the coming decade, I think. And one area we're interested in, working with James Dahlman at Georgia Tech, is to use his nanoparticle technology to deliver to the lungs of animals, and ultimately we hope to deliver functioning copies of that gateway that I've shown you to the lungs of people as well. And then I just want to say finally, and I've said this probably enough already, but the technologies I've described to you here today for cystic fibrosis, whether it's understanding molecular mechanism, whether it's using robotics and high-throughput compound library screening to discover new therapies, or whether it's implementing those therapies in a personalized way, those approaches have ramifications far beyond CF. And so for many other genetic diseases, this is an example of a disease called adrenoleukodystrophy. It's the Lorenzo's oil disease, which was discussed a lot in a movie that Nick Nolte and Susan Sarandon were in many years ago. This disease is caused by a protein that looks an awful lot like the cystic fibrosis protein when you study it. And so it opens itself up immediately to a screen of hundreds of thousands of drugs to find drugs that will overcome this problem as well. And I think in the future, we're going to see many diseases managed in this way. So then to conclude, I'd say substantial progress has been made towards development of novel therapies that address the most fundamental problems that cause CF. And that although we used to try to group all the mutations in categories, Many are complex, and so things are a little less precise than precision medicine initially envisioned, but things are working very well across many subcategories with many different drugs that are in the pipeline. The cost and patient access can be challenges, but there are creative ways that are being used to approach these issues, and they will continue now in the future to be challenges that need to be overcome. And then to say that this high throughput compound library screening offers hope towards treatment of numerous disease states. And these are collaborators at Emory, collaborators at the University of Alabama at Birmingham, where I worked for many years before coming to Emory. For some of that I often discuss groups in Germany and also at McGill. And then to the groups that fund our research, none of this would be possible without the people in the audience today. Our funding comes from the National Institutes of Health, from Emory and Children's, from the Cystic Fibrosis Foundation and Georgia Research Alliance. And I thank you very, very much for your attention, and I'm glad to answer any questions you might have. And it's a pleasure to be with you here from Atlanta. Thank you.
Thank you, Dr. Sorscher. That was so informative, and really appreciated learning about your transformational research and how it's being used to create these these personalized and tailored therapies for people with CF and other diseases. We do have a question from the audience that says, what are the chances that myself or my partner are carriers? And would you recommend getting genetic testing to find out if we are carriers?
It's a great question. The carrier frequency among people of European descent, Caucasians, it's something on the order of 1 in 30. It might be a little higher or lower than that, depending on on your ancestry. It's a simple thing to be tested. If you're thinking of having children, it can be done pretty routinely, and it's a personal choice, of course, for a couple to make before they have children. But it's often something that's undertaken.
Great. Well, thank you for sharing that. We did have some talk about genetic testing, and I think that is something that people are beginning to look at more and more. We have Jennifer Kyles asking, how is CF different from asthma? They sound sort of similar.
Jennifer, I just say that's a great question, and it's one we've been interested in because in asthma, as you know, there are problems with breathing. It can be very tight. It can be like having someone's hands over your face, just like CF. And there are also thick mucus plugs that can come up. Sometimes infection, although not as frequently as in CF, but many similarities. And another disease, chronic bronchitis, also has some of these same manifestations. There is some information that the same protein that causes cystic fibrosis may sometimes contribute to a disease like asthma or like bronchitis. And that work is still being pursued, so it's not completely worked out. But there are several laboratories interested in whether some of the same drugs that work for some of these other conditions that work in CF might have a place in other conditions, other diseases of the lung. I think it's too early to say anything for sure, but it really is an insightful and good question. And it's gratifying to hear because it is at the very cutting edge. So the goal of the presentation was to bring everyone to the leading edge of what we know, and it certainly is a cutting edge question.
Great. And besides Emory, if someone does have CF and they would like to have this high throughput robotic screening, where else can they go? Is this offered to, at hospitals all around the country?
This is a technology that isn't done on a person by person basis. It's a technology for a whole category, say, of mutations in the cystic fibrosis gene, and it's typically done at big academic centers. There are academic centers that run these tests, and also many commercial groups and some non-for-profits. So the kinds of screens that are run are usually run based on numbers of patients that might have the form of a disease, and also the ease with which the assay can be developed. It's not the kind of thing where if you say, I have this mutation, if you were the only person in the world with that mutation, it's unlikely that 1 million compounds would be screened just for you. It's more likely you would be grouped with other people that have a similar kind of CF or a similar kind of disease, and the compounds might be screened against people with that form of the disease. Yeah.
Great. Well, thank you so much, Dr. Sorscher. That was so fascinating and informative. We really appreciate you joining us today, and we appreciate your leadership at Emory University. Before we end the program, I do want to remind our listeners that all Emory Alumni Association programs are posted on our website, at alumni.emory.edu, and you can also find podcasts of all of our faculty lectures on iTunes U at alumni.emory.edu/faculty on iTunes. So this concludes our faculty webinar for today, and we appreciate you all for joining us. Thank you for supporting the Emory Alumni Association, and have a wonderful rest of the day. Thank you, Dr. Sorscher.
Pleasure. Thank you for having me.